All right, this is part 2 of the lecture on the basics of DNA and cell division. This will correlate with chapter 3 in both the Explorations and Essentials textbook. Mitosis is a type of cell division that will produce somatic or body cells. So it begins with DNA replication followed by one cell division. It occurs in all somatic cells, all body cells throughout the body, so skin cells, organ cells, tissue cells, blood cells. The end result is two identical diploid cells, and daughter cells will contain a full set of chromosomes. So all somatic cells, just to review, will have 46 chromosomes in humans. The first phase is called interphase. Interphase you can think of as kind of the prep phase or pregame for mitosis. During interphase, the big events are DNA replication and also the replication of organelles. During prophase, the DNA will condense into visible chromosomes and the spindle apparatus begins to form. During metaphase, all of the sister chromatids will be aligned across the equator of the cell. So a good way to remember it, meta equals middle. So the middle phase, all of the chromosomes are aligned across the middle or the equator of the cell. During anaphase, there's going to be a process called disjunction. So the spindle apparatus is going to rip apart the sister chromatids at the centromere and pull the now individual chromosomes to opposite poles of the cell. And then the conclusion is called telophase or cytokinesis, where the nuclear membrane will pinch in and form the two identical diploid daughter cells. So mitosis begins immediately after an egg is fertilized. So from zygote formation all the way through death, your cells are going through mitosis. Some cells are constantly going through mitosis, like your skin cells, for example, because they're exposed to the environment. They're going to be constantly going through mitosis for growth and tissue repair. Cell division continues and cells become specialized to perform specific functions through the process of differentiation. So some somatic cells become tissues, some become organs, some become blood cells, bone cells. You guys get the picture. All right, so many types of cells, such as your skin cells, are constantly replaced. Other cells, such as nerve cells, are rarely replaced. So that's why if you get nerve damage, it's so detrimental because those cells are not going through mitosis nearly as frequently as skin cells, for example. So in mammals, the entire process will take approximately 18 to 24 hours. But the vast majority of the time, your cells are in the prep phase or the pre-phase called interphase. So that's represented by the red arrow here. The actual division process, prophase, metaphase, anaphase, and telophase, takes about an hour. So there are some video links in the module. So there's an animation of mitosis that I would like you guys to watch after this PowerPoint. All right, meiosis will be what occurs when we're producing gametes or sex cells. So it's one DNA replication followed by two cell divisions, meiosis 1 and meiosis 2. It begins with specialized cells in the gonads of both males and females. We'll talk about that more specifically here in a moment, but it will produce sex cells, so sperm and egg cells. End result is 4 haploid daughter cells. Remember, diploid double, haploid half. So every single sperm cell, every single egg cell contains 23 chromosomes. And because of the process of recombination and independent assortment, the reshuffling of genetic material, it does not result in identical copies. This is the process of sexual recombination. So even if you have the same parent as your siblings, you do not necessarily look the same as your siblings because every single time a gamete is formed, this process of recombination and independent assortment create genetically unique gametes. Let's talk a little bit about meiosis in males. So I mentioned that it begins with a specialized somatic cell that is found in the gonads of males, the testes of males, that goes through meiosis 1 and meiosis 2 to produce genetically unique sperm cells. So that specialized cell is called a primary spermatocyte. In males, the production of sperm cells will begin at puberty and continues all the way through death. Sperm cells are produced and stored in the epididymis for 10 to 20 days, where they're either used or if they're not used, they're reabsorbed by the body. All right, so just like there's a primary spermatocyte in females, there's something called a primary oocyte. So the primary oocyte is a specialized somatic cell located in the ovaries of females that will divide to produce genetically unique egg cells. So in females, the production of egg cells will begin before that female is ever born, beginning about the 12th week after fertilization. So both your grandmother and your mother carried you, because when your mother was still in your grandmother's womb, your mother was already producing egg cells. So females are born with about 30,000 eggs, and these eggs are held in what we call suspended animation until that female hits puberty. Once that female hits puberty, she will have the onset of menstruation, which is called menarche. So the onset of menstruation is the beginning, and then an egg is released every 28 days. The fertile days are usually the middle of the cycle, about day 12 through approximately day 16. So if the egg is fertilized, then it eventually becomes a zygote and then eventually a developing fetus. If the egg is not fertilized, then it's released from the body through the process of menstruation. So for females, there's more of a marked beginning and end to the process. It begins at menarche, or the onset of menstruation, and will continue until menopause. All right, so here's the link. There's another video clip I want you guys to watch on meiosis. So the link is going to be within the Canvas module. All right, so this slide here is just showing mitosis and meiosis right next to each other, so you can compare and contrast. So mitosis is the production of identical somatic cells. The function, why we care, the function is growth and tissue repair. So without mitosis, we wouldn't be able to replace our skin cells when they get damaged, or organ cells, or bone cells, muscle cells. Growth and tissue repair is very important. So the end result, two identical diploid cells. So remember, diploid in human means in every nucleus there are 46 chromosomes. Mitosis occurs throughout the body, beginning at fertilization or zygote formation and then ending at death. Meiosis is there's two parts. There's meiosis 1 and meiosis 2. The process of recombination and independent assortment, the reshuffling of genetic material, will produce genetically unique gametes. So the function, why we care, it's obviously necessary for reproduction and genetic variation. The end result, 4 non-identical haploid cells. So remember, if it's haploid, it means half. So half of 46 is 23. Every sperm cell, every egg cell contains 23 chromosomes. It occurs in the gonads of males and females. So remember, there's the primary oocyte located in the ovaries that goes through meiosis 1 and meiosis 2 to produce genetically unique egg cells. And then the primary spermatocyte will go through meiosis 1 and meiosis 2 to produce genetically unique sperm cells. All right, so there are what we call some chromosomal abnormalities or aberrations. This occurs when there's a mistake during meiosis resulting in a chromosomal abnormality. So a chromosomal mutation would be a mistake that results in an extra or missing piece of a chromosome, an extra or a missing chromosome, or even an extra or missing set of chromosomes. So a chromosomal anomaly is having too many or too few chromosomes. So the standard condition is pairs of chromosomes. But for example, if there's a non-disjunction, so remember, during anaphase, that's when the chromatids are ripped apart. If there's a non-disjunction that prevents the 21st chromosome from separating, that means that you will have either too many or too few chromosomes. So trisomy 21 is when you have a third 21st chromosome. So the more common name for that is Down syndrome. Chromosomal aberrations, so if you remember back to the slide when we saw the karyotypes, so the larger the chromosome, the more genetic material, genetic information that's located on that chromosome. So if there's an aberration or mutation on the chromosomes that are larger, sometimes that's too severe for survivability. So that can result, basically the body recognizes that the fetus is not viable, and it can result in an early miscarriage. All right, so non-disjunctions occur when the chromosomes fail to separate properly during cell division, during anaphase. So if a non-disjunction occurs during meiosis, then the resulting gametes, egg cells or sperm cells, will have an improper number of chromosomes. So the standard condition, what you see in a typical human, is a disomy. So you break up that word, di means 2, somy means chromosomes. So that's the standard or normal condition. So you have 22 pairs of autosomes and then 1 pair of sex chromosomes. A monosomy occurs when one pair is missing. So an example of that is XO syndrome or Turner syndrome. That's still biologically a female, but she will have some genetic abnormalities because she has only one X chromosome and not a second X chromosome. A trisomy, so break that word up. Tri means 3, somy means chromosome. That occurs when there's an extra chromosome present. So if a non-disjunction occurs at the 21st location, you've got a trisomy 21. If a non-disjunction occurs at the 11th location, you've got a trisomy 11. If a non-disjunction occurs at the 18th location, then you've got a trisomy 18. All right. All right, so let's talk about different types of genes. We will get into this in more detail when we work with Punnett squares and also the Hardy-Weinberg equation. So just like there's types of proteins, there's also types of genes. Structural genes are responsible for body structures, and regulatory genes turn other genes on and off. So that's essentially how our body knows how to create various structures. So homeotic or Hox genes, those are turn various things on and off, and master genes do the same. A polymorphism, so each gene has a specific physical location, so it's always represented by a band on the chromosome. So for example, here we're looking at the color of flowers. So alleles are variations of these genes. So if they are chemically the same, then you call them homozygous. If they're chemically different, then you call them heterozygous. All right, so let's talk about genotypes and phenotypes. So just like it sounds, a genotype is the genetic makeup of an organism. So on the homologous chromosomes, if you receive a dominant allele from both parents, you're always going to represent a dominant allele with an uppercase letter. So if you have 2 uppercase letters, that's representing a homozygous dominant genotype. If you receive a dominant allele from one parent and a recessive from the other, you're going to call that heterozygous. Chemically different alleles are called heterozygous. If you receive a recessive allele from both parents, from mom and dad, then you're going to call that genotype homozygous recessive. The phenotype is the physical expression, and it may also be influenced by the environment. So the phenotype is how the organism physically looks. So we'll look at traits like earlobe form, tongue rolling, whether or not you have a widow's peak. Those are some of the basic Mendelian traits that we'll look at to understand the basics of genetics. So just to review, chemically identical alleles are called homozygous. So you break that word up. Homo means same. Zygous means zygote. So if you get a dominant allele from both mom and dad, then you are homozygous dominant and you represent it with 2 uppercase letters. If you receive a recessive allele from both mom and dad, then you are homozygous recessive. If you receive a dominant allele from one parent and a recessive from the other, then your genotype is heterozygous for that trait. All right, so human blood type is an example of a polymorphism. So human blood type is a genetic trait with different alleles. Because there are more than one dominant allele, or there's more than one possibility. So in humans for ABO blood typing, both the allele that codes for blood type A and the allele that codes for blood type B are dominant. If you have an AB blood type, then it's codominant because both will be expressed in the genotype. And the only way you end up with O is the recessive. You must receive an O allele from both mom and dad. So the combination on your homologous chromosome is going to determine that person's blood type. So there's 2 different genotypes for blood type A. If you receive an A from one parent and an A from the other, then your blood type is blood type A. If you receive an A from one parent and an O from the other, you're still blood type A because that A is going to be dominant over the O. Same deal for blood type B. There's 2 different genotypes for blood type B. You could either receive a dominant B from both mom and dad, or you could receive a dominant B from one parent and a recessive O from the other. If you receive an A allele from one parent and a B from the other, that's the codominant situation. That's blood type AB. And if you receive an O, recessive O allele from both parents, then you're blood type O. When a person receives a blood transfusion, the transfused blood must have the same blood type as the recipients in order to avoid what we call an antibody-antigen reaction. So what happens if you have blood type A and you receive a transfusion from someone with blood type B, your body is basically recognizing that as foreign. So there's going to be this antibody-antigen reaction that's going to cause your red blood cells to clot. And that will mean that you can't get proper oxygen to all of your organs. Your red blood cells are carrying oxygen to all your vital organs. So when the wrong blood type is given to a person, the body's immune system recognizes these antigens as foreign, and it causes this process of agglutination or blood clot. The antibodies attach themselves to the foreign antigens, causing clumping of the red blood cells, and the clump red blood cells will be unable to pass properly through the blood vessels and carry oxygen to your vital organs. All right, let's talk about polygenic variation. So most of genetics is based on this one gene, one protein model. We call these basic Mendelian traits. However, especially with humans, many traits are polygenic and determined by genes at more than one locus or location. So polygenic traits are determined by genes at 2 or more loci, different locations on chromosomes, and it can also be influenced by the environment. For example, skin color, skin pigmentation is determined by at least 3 separate genes, and it is also influenced by environmental circumstances. We'll talk more about that in Chapter 5. For some traits, only some of the genetic variation can be calculated. We call that heritability. So the heritability ranges from 0, meaning none of the variation is genetic, to 1, meaning all of the variation is genetic. And only traits that are inheritable will respond to the force of natural selection. All right. Most complex traits are both pleiotropic and polygenic. So just remember, polygenic refers to one phenotypic or physical trait that is affected by 2 or more genes. And the physical manifestation is influenced by environmental circumstances, such as UV exposure, climatic conditions, etc., etc. So some examples will be skin color, height, head form, tooth size, and eye shape are all polygenic traits in humans. Pleiotropy is referring to one gene that can affect more than one phenotypic or physical trait. So it's a single allele that's having multiple effects. All right, that's the conclusion of this PowerPoint presentation.